It's a slow Monday, I guess. Monday evening. Well, hello, everyone who's here. Let's start. There's a couple of things that I want to do today. One is to ask you if you've noticed the new assignment on cell division.
The one for extra credit?
Yeah, this is an extra credit. If you haven't noticed that, look in assignments and find it. And I described it a bit in the assignment itself. It turns out that the biology department here at Quimaca has an obligation to report what are called student learning outcomes, SLOs. And they've chosen the topic cell division to assess that in this section. So what we've been provided is a set of 54 questions, and you'll get credit for that, for each one that you get right. And it's due on the 28th, I believe it is, 27th, due on the 27th. And then we'll discuss it in class on the 28th. It's a good learning exercise to find out just how good a command of the topic we have.
Professor, I have a quick question. So is that okay to get it wrong? To answer like wrong questions?
Yeah,
Okay.
yeah, you do the best you can. But if you try to do one and get it wrong, that's okay.
Mm-hmm.
But I should emphasize that this is open book in contrast to most of the exercises that we have. This one's open book, so you can use any resource, your notes, the slides, the recordings, the internet, talk to people, whatever you like, you can use to answer the questions. So the combination of that, plus the fact that we've already gone over this quite extensively, I would expect that this section will do very well. Any other questions about that special extra credit assignment? You have another worksheet that's been assigned, enzymes and catalysis, and that is due tomorrow. And we'll discuss it on a Wednesday, in class on Wednesday. And I would like to review quiz 7, which you just took last week. How do you see it? Document, quiz 7, cell division and Mendelian genetics.
No, I see it's like a Zoom meeting or something, link.
Okay, I'll have to start over. Now, how about that?
Yep, we're good.
Good? Okay.
Yes.
I want to go over each of the questions in this quiz. The average grade was 68. The high grade was 100. Several people got that, but the fact that the average is still quite low indicates that a lot of people didn't. And that's my concern, that you understand for the questions that you didn't get right, understand why and why that question was asked. So the first one, the goal of mitosis is blank, and the goal of meiosis is blank. Only about 9% of the class got it right as I expected. And what by right, I mean the goal of mitosis is exact reproduction, and the goal of meiosis is to introduce variation. So, so most of you didn't get exactly what I was looking for, but many of you got part of it. You would answer with information that wasn't wrong, but it wasn't exactly right either. So if what you put down was not wrong, you got partial credit for it. So most of the class got partial credit for question number 1. And I've indicated here where that information, where from the slide set that information came from. Could somebody mute their mic? I'm getting some feedback here. So in that slide set, there was a slide on mitosis consequences and meiosis consequences. And in the mitosis one, it says no genetic variation is introduced, and it makes 2 cells identical to their progenitor. So that's what I was looking for. Meiosis, genetic variation is created. And then there was farther on in that slide set, an exact comparison between mitosis and meiosis. And right at the top, mitosis goal is exact reproduction. Meiosis goal, introduce variation. Now, those aren't the only things that happen in mitosis and meiosis, but they are, in my opinion, the most important things, and they're the things that I emphasized when going over the materials. Question number 2 is about Mendelian genetics. As I've said before, you need to read these questions carefully. About half of you got this right, but half didn't. So hemophilia is an X-linked disease, causes bleeding. What is the likelihood of a male with hemophilia passing this trait along to his progeny? The first thing you'll notice, probably did notice, is that it doesn't say anything about the female. So since this is a question about hemophilia and nothing is said about the female, then you know that the female doesn't have it. She's not a carrier and she's not sick. So she's normal. Her genotype, since this is an X-linked disease, her genotype is XX. The male, the question indicates, is a hemophiliac. Now, he only has 1 X chromosome. And since this is X-linked, we know that his X chromosome is mutant. So if we look at the gametes, and this is always a critical step in this kind of problem, you look at what kind of gametes are produced. what can the male produce? He has an X star and a Y. So he can have either an X star sperm or a Y sperm. Well, the female we know is XX. So the only gametes that the female can produce are X and X. So if you do the crossing, you see that one of the female progeny is XX star. The other female progeny is XX star. The males are both normal, they're both XY. So the question asks, what's the likelihood that a male will pass this trait along? It's 50%, and all the progeny that get it will be females. Any question about that? About half of you didn't get this right. So if you have a question, any uncertainty how to do this, how to think about it, please speak up. Then we'll go to number 3, which is about the phases of meiosis. Crossing over occurs at prophase of meiosis 1, whereas random assortment occurs at metaphase of both meiosis 1 and meiosis 2. And that comes from the slides in the lecture. The first one, meiosis 1, says explicitly that crossing over occurs at prophase and independent assortment occurs at metaphase. Now, this is the only opportunity there is for crossing over. And I put this other slide here to remind you what crossing over involves. It's when the chromosomes come together very tightly in physical contact with each other, and they form contact points called chiasma, and it's at those points they exchange parts. So you end up with recombinant chromosomes, in this case, red chromosomes with blue tips and blue chromosomes with red tips. And since this only occurs in prophase of meiosis 1, that's the only chance, in answer to the question, this crossing over occurs in meiosis 1, whereas random assortment can occur, if you look at the next slide, at metaphase in either meiosis 1 or meiosis 2. And that's because in both of those cases, in both metaphase 1 and metaphase 2, chromosomes line up in the center of the cell, and then they randomly assort, and they go to either one side of the cell or the other, and that choice is entirely random. And that happens twice, metaphase 1 and metaphase 2. So there are 2 different opportunities for random assortment, only 1 opportunity for recombination. About 10% of the class got all 5 parts of this question. All 5 parts right. Most of you got a partial answer. But again, you should know the phases of both mitosis and meiosis and know what happens in each phase. Then there was a matching question, number 4. The gene that contains 2 identical copies of an allele is homozygous. This next one, it takes a little bit of thought. Someone who is a carrier of a genetic disease. There could be any number of answers to that, but if you look at the list of possibilities, heterozygous is the only one that fits. 1 copy of an allele masks the phenotype of the other copy. That is complete dominance. A set of alleles that governs the expression of a particular gene, a particular protein, is a gene. the crossing of individuals producing a progeny with intermediate or blended phenotype is called incomplete dominance. And finally, relaxed form of DNA is chromatin. The next question, in the cell cycle, what happens in the M phase? Mitosis. Most everybody got that right. And finally, number 6, only about 10% of the class got this right, which surprised me because it's a simple monohybrid cross, which we went over a number of times, how you do this. There's nothing complicated about it. You simply have to pay attention to what the description that you're given and what is the question that's being asked. So in this case, the description says that both plants with orange blooms and blue blooms are true breeding. That means that they've gone through a series of inbreeding generations such that they're both homozygous at all loci. You also know from what the description says that orange is recessive. So the first thing we have to do is give the gene that's controlling this color a symbol, and let's call that B for blue. So we know that since orange is recessive, the orange genotype is little b, little b. Therefore, blue is dominant, and we give it the genotype big B, big B. The parental cross then, where we cross an orange one, an orange inbred, and a blue inbred, is between a homozygous dominant and a homozygous recessive. So you know then that by definition, the F1 is going to be all heterozygous, all big B, little b, which means they'll all be blue. So that's the answer to the first question. What phenotype will you see in the F1 generation? It's blue. Now to get the F2, you know how to generate an F2. You cross two F1s. The F1s are all identical, so they're all heterozygotes. You cross a heterozygote, big B, little b, with another heterozygote, and you can draw out a Punnett square to see exactly how it works. And if you do that, you'll see that there is a big B, homozygous dominant. There are two heterozygotes and one homozygous recessive. So the heterozygotes and the homozygous dominant one are all blue, and only the homozygous recessive is orange. So that means the answer to number 2 in the F2 generation, 75% of the phenotypes are blue and 25 are orange. So a lot of you had trouble with this question, and I would like to know if you're still having trouble with this, let me know. Let's talk about it. Both for 2 reasons. One is you need to know how to do this kind of problem. And number 2, it's coming up again in the test 3, which is coming up a week from today. You'll be asked this kind of question, probably multiple times. And in the final exam, I'm sure this will come up again. So you need to know how to do these, how to think about it, how to ask what are the gametes, and then how to generate the products of the gametes.
Professor, I have a quick question for you. So the question is, what are the phenotypes? But you didn't ask us about the genotypes.
That's right.
So, did you want us to include the genotype in our answer or just the phenotypes? I think I was confused about that.
All I wanted was phenotypes. So I specifically only asked for phenotypes. If you put genotypes down, I didn't penalize you for that, but you didn't.
Okay.
But you raise a good point, and that is read the question carefully and answer the question explicitly. You don't have to answer more, but you shouldn't answer less either.
Okay, so I just want to make sure. So when you're talking about phenotypes, you want the percentage and the physical description, but not just the physical description.
It doesn't even ask for ratios. It just says,
Okay.
in fact, I think there was one person who answered with 2 words. Blue, well, 1 word for the F1 and 2 words for the F2, blue and orange. And that answers the question.
Okay, because I think I answered that way and I got partial credit. So I can
Okay.
come back to that.
So let me look that up. If you answered that way, you should have gotten full credit. Let me write that down. And I will check on it after class.
Okay, thank you.
Anything else on the quiz? Test is a week from today, and that will cover cell division, mitosis and meiosis. It will cover dynein. Remember the project that my student did on motor proteins. It will cover Mendelian genetics, probably be heavy on that, and energy and enzymes, which is the topic for today.
Professor, is that going to be on Monday or on Wednesday?
Monday, the 26th. Monday, the 26th, a week from today.
Okay, thank you.
You see the slides? Overview?
Yeah.
Yes.
Okay. I'll show you this overview slide every time we're in this section of the course because I want to keep your minds focused on the topics and the relationships between them. We're talking about energy, enzymes, photosynthesis, and respiration. When we talked about the second law of thermodynamics, we know from that we need a constant input of energy to keep living things alive. So what is energy? Where does it come from? And how do we use it to keep ourselves alive? What are enzymes? What is their structure? What is their function? And importantly, how do they act? How do they act to reduce the activation energy of a reaction? And critically, how are they regulated? Our bodies are full of enzymes. Thousands of reactions are going on at all times. And if all the enzymes were running at full speed, it would be unsustainable. So that means we want enzymes to function only when they're needed and the rest of the time shut down. And we're going to talk about that today. How do they do that? And then finally, both energy and enzymes are involved in both photosynthesis and respiration. Photosynthesis is the capture of energy from sunlight and converting it to chemical energy. And respiration is the opposite. It's how we derive energy from the food that is produced by photosynthesis. I'm going to skip because we've gone through all of this before. I'm going to skip through and only talk about points that I want to emphasize. One is this electromagnetic spectrum. You recall that when you go from very long wavelengths to very short, in approximately the midpoint of that range, there's this clear area called visible light. That's the light that we can see with our eyes. And it's the range of radiation that plants can perceive. Plants can see red, yellow, and blue. By see, I mean they can absorb those wavelengths. They cannot absorb green. So they reflect it. And that's why when you look at most plants, they appear to be green. However, not all plants are green. Now, I want you, you already know that, and I want you to know why. Well, there are a number of reasons. The first one is if you see, for example, a red plant, and I have here a picture of a red maple. It is a red plant, but it's also a green plant in that it has chlorophyll, which functions perfectly normally. It's just that the chlorophyll is masked by a pigment called anthocyanin, which is red, and it reflects more light than the chlorophyll does, so the plant appears to be red. Many ornamental plants overproduce anthocyanins, and when you go around San Diego, you will see a lot of ornamental plants. You'll see a lot of them that do not appear green, but they really are. Now, another reason you might see a plant that isn't green is because it has a nutrient deficiency and it's yellow, called chlorotic. And here's a picture of a chlorotic plant. Another reason a plant might not be green is that it's an albino. Albinism is controlled by a single gene mutation. Here you see two corn seedlings. One is normal green and the other is albino. Now, this little seedling is growing just as fast as the wild type is growing because it's using nutrients from its seed. But as soon as those nutrients are exhausted, it will die. It simply cannot make any food on its own. However, if you feed it sugar, it will grow and live just like a wild type plant. So the only thing it's missing is the ability to make sugar from sunlight. I doubt if you'll see very many albino plants because who would go to the trouble to propagate such a thing? They usually only occur in laboratories. Another reason a plant might be not green is that it's deciduous. That is, it loses its leaves in the fall. When there are environmental signals just right toward the end of the summer, plants start to degrade their chlorophyll and replace it with colored pigments. So this, for example, is an oak tree in autumn. You can see that most of the leaves on the tree are orange, and many of them have fallen to the ground. So this tree is about to shed all of its leaves and get ready for the winter. So those are some of the reasons that you might see non-green plants. There's one other, and I think you've probably all seen this and probably didn't recognize what it was. This picture was actually taken in the desert east of San Diego. What you see is a green plant, a desert plant, and there's this yellow part appearing on the plant. That is a parasitic plant known as dodder. If you look at it more closely, you can see that the dodder grows as fibers, little strings that are associated with the green plant. Now, the dodder is incapable of making any chlorophyll. It cannot make its own food at all. It gets its food from its host plant. You see here in the lower left how it does that. In this case, it's wrapped itself around a stem of the host plant. And you see these little protrusions called haustoria, which actually invade through the epidermis of the host plant so that they can soak up nutrients. And on the lower right, you see a photomicrograph of a host parasite interaction. You can see the host cells at the bottom, parasite is at the top, and the haustoria have invaded into the host where they have access to nutrients, access to everything they need, including water, minerals, and sugars. So a parasitic plant such as this doesn't immediately kill its host, but if it stays around long enough, it will. That's why it's called a parasite and not mutualistic, because it really provides nothing for the host. The host provides everything for it.
Professor,
Yeah.
is that different than like dry brush?
Yeah.
Okay.
It's different from dry brush.
And then the daughter, would it can only survive if it has a host, correct?
That's right.
Okay.
If you take it off the host and throw it out on its own, it'll die. But it's,
Does it need water?
it gets water from the host
From
plant.
the host. Okay.
So, Professor, I'm sorry, this is Taylor.
Uh-huh.
So does the daughter also refract the light, or just the host?
The daughter, it's a yellow color, which means that it's reflecting yellow light and it's absorbing everything else. But it has no pigments that can convert sunlight into energy. So it's completely helpless without its host.
Okay, so since it's not part of the host, that's why it's yellow and not green.
It's not green because it has no chlorophyll.
Gotcha. All right, thank you.
Uh-huh. Anything else about parasitic plants? I'd encourage you when you drive around, both in town and in the countryside or in the deserts, to keep your eye open for these various different colors of plants. We just all expect plants are going to be green, and if they're not, we ignore them. But when you go to the desert and you see a yellow patch, now you know what it is, and you know the significance of it. And as you're driving around in more urban areas, if you see an ornamental plant, now you know it's not without chlorophyll. It has chlorophyll. It's just got another pigment instead, or as well. And you also know that disease can cause plants to be not green. Mutations can cause it, and it's natural in the fall for leaves of deciduous trees to lose their green color. I want to page ahead again to the next important point. And this is it. Contrasting endergonic and exergonic reactions. Endergonic require energy input. Exergonic release energy. And a good illustration of this is to compare photosynthesis with respiration. These are two topics we'll look at in detail in the future. But for right now, let's use it to illustrate endergonic and exergonic. In photosynthesis, you start with carbon dioxide and water. Energy in comes from sunlight. And that process ends up making glucose and oxygen. Now, the reverse of that is the way we derive energy from food. If you start with glucose and break it down through respiration, out comes energy along with the starting materials, carbon dioxide and water. And that is an exergonic process. Photosynthesis is endergonic. Remember activation energy. It's the amount of energy that it takes for a reaction to proceed, and an enzyme reduces that. And then I want to talk about the mechanism by which an enzyme reduces the activation energy. And I've chosen this slide to make the point. We went over this last time, but I'm re-emphasizing it now. If you look at the upper left, enzyme is yellow, substrate is green. The substrate fits into the active site of the enzyme. The enzyme then can change shape such that it either breaks or weakens the bonds in the substrate, so that when the enzyme releases it, there are now two products made from the substrate. So this is something that's very important for you to know. We'll probably ask you something about that at some point. On this slide, I mentioned that you should know what a reaction rate is. It's the speed with which a substrate is converted to a product. And what this slide shows is that the reaction rate with an enzyme is much faster than without.
So Professor,
Yeah.
with the reaction with an enzyme, it seems like it tends to like mellow out or plateau. Does that one on the bottom eventually mellow out, or is it always a sharp line going like that?
When it reaches the maximum rate, it stops, it plateaus. In enzyme kinetics, that's called the Vmax. So every enzyme has a maximum rate, maximum speed that it can work. And once it's reached that, it won't go any faster. It doesn't matter what you do to it, it won't go any faster. And without enzyme, yeah, the reaction would proceed. And if you waited half your life, it would probably get to the Vmax. But for practical purposes, you wouldn't have to consider that. So with an enzyme, Vmax is usually reached fairly quickly.
Hold on.
A question? If not, mute your mic. I want to emphasize a point about denaturation, because that's what happens when the environment is not conducive to enzyme activity. So what are the things that cause denaturation? Well, some of them are listed here: pH, temperature, salt concentration, solubility. So what I want you to keep in mind is that proteins are not just globs like you see here, this blue glob. They're these three-dimensional structures that are highly complex, folded and cross-linked with various different kinds of bonds. And they have to be in precisely the right configuration to be the right shape for a substrate to fit in there. And in a minute, we're going to come to inhibitors. There has to be a place in the enzyme for the inhibitor. Now, if this intricate molecule is abused, it will denature. And under extreme conditions, it will completely fall apart so that all that's left is the primary structure, just a long chain of amino acids linked together with peptide bonds. And in this denatured state, this protein would have no enzymatic activity at all. Now, some denatured proteins, if you take away the extreme condition that's caused the denaturation, they can renature and go back to their original configuration and have enzymatic activity again. But usually, yes?
I had a question about when it's when the protein's denaturized, does it like change the shape or like does it look different after?
Yeah, this shows.
Yeah, like okay.
Looks
So
like,
even like yeah. Okay.
so it could be partially denatured. Doesn't have to denature very much. All it has to do is lose the pocket, say for the active site, and it will no longer function as an enzyme. It won't convert substrate to product, but it may not be completely denatured at that point. In which case, if it's slightly denatured, it has a better chance of renaturing. But if it's completely denatured like this one, the chances are not good.
So Professor,
Uh-huh.
you were saying before that when it denatures that it'll like cut it up into pieces and put it back into another enzyme. Wouldn't that cause the next enzyme to be damaged too if it gets part of that damaged piece from the previous one?
Denatured proteins are not cut up into pieces. Well, I think I know what you're referring to. Yeah, so someone asked what happens to the denatured protein? And usually what happens is that it's degraded to its constituent amino acids, and those amino acids are recycled. They go back into making new proteins. So given that, then what is your question?
So if it goes into the new amino acid and part of it was damaged, what if it gets that damaged piece? Would it cause the next enzyme or amino acid to be damaged because it got that piece that was messed up?
Yeah, very good question. The answer is no, in a word. So if a denatured protein, like you see here, that's a series of, say, 1,000 amino acids. And if you take those apart, those amino acids are all just fine. They're not affected. Amino acids are more stable than proteins, and so they're not affected by the high pH or the high temperature. And they can go into a vesicle and get transported to another part of the cell, say to the endoplasmic reticulum, and can be recycled. They can go back into making a totally new protein, but the amino acids themselves have not been damaged. So in the new protein, there's a different sequence of amino acids, but the amino acids themselves are authentic. They're not damaged. I hope that's clear. If it's not, say so, and I'll try to explain it again.
No, I understand now.
Okay.
So, so Professor, what you're saying is in denaturization, the proteins break down, but the amino acids themselves are still perfectly fine where they can go and be recycled into something new, right?
Uh-huh. You got it.
Okay, just making sure.
Yeah, you got it. Stated it exactly right. Okay, anything else? Then we'll go ahead. How can the rate of an enzyme reaction be changed? Well, there are three main ways: changing the temperature, changing the pH, or changing the ionic condition of the environment. What you see in this picture are examples of temperature change and pH change. And be sure you know how to read graphs, because you will be asked on tests how to do it. And what you see at the top here is the effect of temperature. The human body is about 37 degrees. And if you look at most enzymes from a human, they would have an optimum reaction rate at 37 degrees. That is, they're going as fast as they can at 37. If you go either above or below that, the reaction rate slows down. And if you take the case of the first curve here, if you got to 50 degrees, the enzyme is denatured, has no activity at all. Now that's true of most parts of the human body. There are some organisms, like heat-tolerant bacteria, live in very hot environments, like hot springs, where the temperature is near boiling. Well, they have to have heat-tolerant enzymes. So there's one that we'll talk about later, Taq polymerase, which has an optimum of 75 degrees. For most any human enzyme would be totally inactive at that temperature. But this one has its optimum at that temperature. And even at 95 degrees, near boiling, it's not very active, but it doesn't denature, even at 95 degrees. And that's a very important feature of this enzyme, which we'll talk about when we talk about molecular genetics. So that's temperature. pH. The stomach, human stomach, has a pH of 2. There's an enzyme that works in the stomach called pepsin, and it has a pH optimum of 2. So if you get up to pH 3.4, which is still very acid, it denatures. In contrast, a more typical human enzyme would be trypsin, another protease, which is active in the intestine at between 6 and 7. Now, you might wonder, why is the stomach so acid? And pH 2 is almost like pure acid. Why do you think that might be?
To break food down?
Yeah. So for example, proteins, under highly acid conditions, proteins will start to, the bonds will start to break. And so they'll start to break down at low pH. So that's one reason. What might be another really important one? Think of the food you eat, where it comes from. Is it sterile?
No.
Right, so what might be in it?
Bacteria.
Bacteria, pathogens of all kinds. So any bacterium that goes into a pH of 2 is dead. So we essentially sterilize our food just by swallowing it. There are other reasons why the pH of the stomach is low, but the main ones to remember are what we've talked about. I want to just quickly go over ATP and ADP because this interconversion of the two is critical to energy metabolism. And we went over this last time, but adenosine is these two molecules, adenine and ribose, hooked together. If you add a phosphate to the ribose, it's now AMP, adenosine monophosphate. Add 2 phosphates, it's ADP. Add 3 phosphates, and it's ATP. And the significance of this is that this last bond, phosphate bond, between the second and the third phosphates is high energy. And when it takes a lot of energy to make that bond, but when it's broken, it releases a lot of energy. And it's that released energy that keeps us alive. You can think of ATP, ADP as a toggle switch. They're constantly being interconverted one to the other. And the point is that the energy going in to making ATP is less than the energy that comes out making ADP. So if you start with ADP and a phosphate group, it takes a lot of energy to make this. But when you do, you have this high energy bond, it's ATP. When energy is needed, this bond is broken. And you go back to ADP and PI, but you release energy at the same time. And we'll talk about where this energy comes from in a little bit. Metabolic pathways are of 2 types, anabolic and catabolic. Anabolic means build up. So you start with small, simple molecules and build them up into more complex ones. Anabolic reactions, anabolic pathways require free energy from the environment. Catabolic pathways are the opposite. Catabolic tears down. So you start with complex molecules, break them down into simple ones, and that the process of breaking those bonds releases energy. So catabolic pathways tear things apart and release energy in the process. This picture illustrates that in very simple form. An anabolic pathway builds up. You take small molecule monomers plus energy to hook them together and make a polymer. So energy then is stored in the bonds of this polymer. Catabolic pathways take this polymer, which has energy stored in it, breaks it apart into monomers and releases energy. At the bottom here, you can see a sort of an overview of how that happens. Think about what goes into your mouth when you eat. It's primarily proteins, polysaccharides, and lipids, all polymers, long chain polymers. And through a long process, which we'll talk about when we talk about respiration, these polymers get broken down into small molecules, which go into what's called a citric acid cycle, and that's where ATP comes from. So you start with the food you eat, and you end up, after a long convoluted process, you end up with ATP, which is the energy that the cell uses to do its work. All these pathways can be reversed, and when they are, they now go from catabolic to anabolic, where they use energy to make high energy polymers. Photosynthesis and respiration, as I keep saying, we'll talk about these in detail later, but these illustrate anabolic and catabolic pathways so nicely because they can be boiled down to something very simple. In the case of photosynthesis, you take two simple molecules, carbon dioxide and water, and you take energy from light and you get glucose and oxygen. That's an anabolic process. It takes energy, and that energy comes from light. Respiration is a catabolic process, because it starts with a high energy molecule, glucose, and oxygen, breaks them down to carbon dioxide and water. These are the starting materials for photosynthesis. But when it does that, it releases energy. And that's the energy that we live on. Now, I want to emphasize, we're running short on time, but I want to quickly go over enzyme regulation, because this is critical to understanding why we don't just burn ourselves up. Okay, enzyme inhibition. There are two types of reversible enzyme inhibitors, competitive and non-competitive, both illustrated here. Now you see the enzyme is illustrated like a blob. The substrate is a yellow blob, and the competitive inhibitor is a blob that's shaped very much like the substrate. So if the competitive inhibitor and the substrate are together in the presence of this enzyme, either one of them can bind to this active site. If the competitive inhibitor gets there first, it occupies the site, and the substrate cannot sit down there, and there's no reaction. So in this case, in this way, the enzyme reaction is prevented. A non-competitive inhibitor works in somewhat the same way, but in another way, it's different. In this case, the inhibitor here in green does not compete for the active site. It has its own site at a different place in the molecule. If it binds to its site, what happens? The shape of the enzyme changes. So now, instead of being open and fitting the shape of the substrate, it doesn't have that shape anymore. The substrate cannot bind there, and the reaction is inhibited. So these are two ways that an enzyme can get slowed down or stopped. So you can imagine if the this competitive inhibitor, if the concentration of the inhibitor was 100 times higher than that of the substrate, then there'd be very little chance of the substrate ever getting into this active site. And the reaction would be almost completely stopped. Now, this is my favorite form of regulation. It's called end product inhibition or feedback inhibition. So first, let's consider a pathway, and this can be any pathway where you start with the substrate, this blue box. Enzyme 1 converts it to another box, enzyme 2 to to another box, enzyme 3A4, and enzyme 5 finally converts it to the red box, which is the end product. Now, supposing this end product is a protein involved in scab formation. You cut yourself, you need to repair the wound and make a scab. So if you do that, there'd be a huge demand for this protein, this end product. And so this pathway then, starting with the blue box, is going to run very fast, making as much end product as possible to heal your wound. But now as your wound heals, the demand for this protein, the demand for making scabs, is reduced. You don't need so much of this. And once you're completely healed, you don't need any of it. But yet the pathway is still running. So this product is being made, it's not being used, so it accumulates. And as it accumulates, it turns out that enzyme 1 has a binding site for this end product. So as this accumulates in the cell, it starts binding to this enzyme and shuts it down. And when this enzyme shuts down, the whole pathway shuts down. No more of this end product, no more of this scab protein is made, and you're not wasting energy making something you don't need. So is that clear how this works? End product inhibition. If the end product gets too high, it has a site in the first step of the pathway. If it can stop the first step, it stops the whole pathway. Clear? And now the last topic is redox reactions. I'm going to read these statements because they're very significant, and they'll become more meaningful to you when we start looking at pictures. But first of all, energy is harvested through electrons that are moved from one molecule to another. Electrons carry energy, some of which will be transferred to intermediate molecules on a downhill journey. And as they go through this journey, the energy is transferred to molecules, but also some of it is released. Well, some substances more strongly attract electrons, and think of electrons as energy, than others. A substance that loses one or more electrons is said to be oxidized. A substance that gains electrons is said to be reduced. These two processes, oxidation and reduction, always occur in pairs. Remember that. If one happens, the other one also happens. It's known as a reduction oxidation reaction, or redox. Now, to illustrate that with pictures, this is the simplest form of oxidation and reduction. So what you see here are substances A and B. Substance A is reduced. It has an extra electron. Substance B is oxidized. But if A transfers its electron to B, now A is oxidized and B is reduced. Now there is a mnemonic for trying to remember this called oil rig. That stands for oxidation is loss, reduction is gain. So oxidation, the first part here, is loss of an electron. The second part, reduction, is a gain of electron. And now let's look at what's more relevant to us, biological oxidation. So here we have on the left an organic molecule. This could be glucose that has 2 hydrogen atoms in excess. You remember a hydrogen atom is nothing but a proton and an electron. There are no neutrons. And while this molecule, this organic molecule has them, it's in a reduced state. Now, if it transfers these atoms to a carrier called NAD, which is a coenzyme, it's an electron carrier, you now have an oxidized organic molecule and a reduced carrier. In this case, the carrier is called NADH plus H plus, a proton, and that carries energy. So it's reduced electron carrier. Not only is it reduced, but energy is released. And so this happens in successive steps. And this illustrates just one step in a long chain. So we start with AH2, A being, for example, glucose, reduced H2. It can hand its 2 electrons off to NAD, which then becomes NADH plus H plus. NADH can hand electrons off to another carrier, for example, cytochrome. And so that this oxidized form of cytochrome now becomes reduced. And just imagine this going on and on. Cytochrome hands off electrons to the next one, to the next one, to the next one, and it goes on down the chain. Now this, it shows you the structure of NAD, nicotinamide adenine dinucleotide phosphate, or without the phosphate, it's just dinucleotide. And this part of the molecule can be oxidized and reduced at will. And when it's reduced, energy is released, H plus. This is very complicated, and just to give you some idea of the magnitude of complexity, there are many enzymes involved here. And if you look at just one of them, NADH dehydrogenase has 45 polypeptide chains. You recall when we were talking about tertiary structure of a protein, one polypeptide chain is highly complex. Here you have 45 highly complex chains all interacting into one huge enzyme molecule. This I put in to illustrate an electron transport chain. The first point is that it occurs within a membrane. And what you have, these big white globs, are enzyme complexes which traverse the entire membrane. So they're embedded in the membrane, and there are actually 4 of them in a complete electron transport chain. And so what happens here is that you start with, say, glucose, from which you get NADH. It hands off electrons to flavin, which hands them off to ubiquinone and to cytochrome B and cytochrome C. And every time there's a handoff, there are 2 extra protons, 2 extra bundles of energy. so that as you keep passing electrons along this electron transport chain in the membrane, protons, ions keep being produced, and they end up on one side of the membrane, so that you're going to end up with, over here, 6 protons. And there's now a charge across the membrane. There are more ions on one side than on the other. And this is where I've talked to you before about chemiosmosis. Chemiosmosis is the transfer of these electrons through the membrane to the other side, where ADP is converted to ATP. This is where we get our energy, from an electron transport chain, which ends up with chemiosmosis. And the final electron acceptor in aerobic respiration is oxygen. Okay, so I hope you have this picture, these huge enzyme complexes embedded in a membrane that are transferring electrons. So what they're doing at every step is this. Every step is a continuous transfer of electrons from one carrier to the next, to the next, to the next. And the whole point of that I'll come to in just a minute. This dramatizes chemiosmosis. Again, you have a membrane, you have these embedded proteins, you have electrons being transferred. As they are, ions are accumulating on one side of the membrane, and then at some point they go through it by the process of chemiosmosis, and ATP is made. Now, this is to show you where in the cell this actually happens. So on the left, this big blue thing is a cell. You can see the familiar organelles. And the little orange things are mitochondria. If you look at one of these mitochondria, you can see that it's really a system of membranes. And it's this inner mitochondrial membrane where oxidation and reduction occur, and chemiosmosis. So it's, after all the complexity of a cell, it's this one membrane within the mitochondrion or the chloroplast where all the action is, all the action for producing energy. And this provides you, I hope, with the logic, the rationale, or even the necessity for redox, for electron transport chains. On the left, you see oxygen and glucose. So you could oxidize glucose and release all the energy at once, and you'd get water and carbon dioxide, but all the energy would be lost. It would be released as heat. And in a biological system, that would do no good at all. The cells would not benefit from a massive release of all the energy in glucose at one time. So what the cell does is slow it down. You start with glucose, and you end up with carbon dioxide here and water way over here. But in between, glucose is broken down step by step with these oxidation reduction reactions, this downhill step after step after step. And at each step, energy is released in the form of electrons. And these electrons continue to accumulate on one side of the membrane until at the very end, they go through the membrane and ADP is converted to ATP. So the point here is that this passing electrons from carrier to carrier to carrier is the way that the process is slowed down and energy can be extracted. So instead of getting all blown away as heat, it's converted into the energy currency of the cell. The cells can use for all the thousands of reactions that are necessary and that require energy. And now I'm keeping you over time, I'm sorry, but just want to finish up with 3 things that we're going to talk a lot more about, and this sort of prepare your mind for it. There are 3 possible ways to make ATP: substrate level phosphorylation, oxidative phosphorylation, and photophosphorylation. And here is a thumbnail sketch of the 3. Substrate level phosphorylation, you start with a substrate, in this case, pyruvate, and ADP, and you transfer the phosphate from phosphoenolpyruvate to ATP. And it's a 1-for-1 or a 2-for-2 exchange. For every ADP, you get an ATP. That's substrate level phosphorylation. And for example, in the next diagram, it's illustrated at the top. The very first step in glucose oxidation is a substrate level phosphorylation. You get 2 ATPs from it. However, oxidative phosphorylation is much more productive than that. So you get these NADH, for each NADH, you get 2 protons. And as it's broken down, and we'll talk a lot more about the Krebs cycle later, the only point I want to make here is that in oxidative phosphorylation, you keep accumulating electrons. And these electrons get fed into the electron transport chain, which is shown here. So that instead of getting 2 ATPs, you end up, after all these passages of electrons from carrier to carrier to carrier, you end up with 38 ATPs from 1 molecule of glucose. So that is very efficient. Photophosphorylation is much like oxidative, except the starting point is different. Instead of starting with glucose, you start with light, which is intercepted by chlorophyll, which ends up giving you excited electrons, which can be passed through the electron transport chain, just like they are in oxidative phosphorylation, and you end up with ATP. And I have one little diagram here of chemiosmosis, which is the most simplified illustration of it, where it's simply a membrane and a high concentration of ions on one side, a low concentration on the other. And as ions move across the membrane, energy is captured. So chemiosmosis is defined as the movement of ions across a semi-permeable membrane down their electrochemical gradient. And the result of that movement is the synthesis of ATP. And I do think that's the last slide. Yes, it is. So we've run over time. I'll try to make up for it some other time. But I'll stop there and ask you if there are any questions quickly before we go. On Wednesday, we'll start discussing photosynthesis, and we'll also go over the worksheet on Wednesday.
Professor,
Yeah,
will we have 2 attempts for this exam on Monday?
1 attempt. It's a full period, 75 minutes, and it's worth 100 points. Anything else?
I have a question.
Mhm.
So what is the passing grade for this class?
I don't know. I'll wait to see how the grades look at the end. I might grade on a percent. I might grade on a curve. It depends on how the grades turn out and what seems to be fair relative to the grades that are distributed throughout the class.
Okay, thank you.
So I can't really answer your question at this point. I can tell you that in the past, I've tended to curve. Okay, anything else before we go? No? Okay, then I will see you all on Wednesday.
Thank you.
Thank you for coming and have a good evening.
You too.
Have a good night.
Okay, bye.